Good evening. In this video, we are going to discuss and explain the light-dependent reactions in more details. So, as you know, there are two photosystems that are responsible for the light-dependent reactions. The first photosystem is that is in action, as we mentioned at the beginning. If we can go backward in order to see what we have said about the stages of the light reactions. So we have two components for the light reactions. We have the light-dependent reactions that result in the formation of ATP and reduced NADPH, and there are the light-independent reactions in which the products of the light-dependent reactions, ATP and reduced NADPH, are utilized. We also talked about the photosystems, and we said that the thylakoid membranes carry two types of photosystems. So we have photosystem 1 and photosystem 2, and both photosystems are a group of photosynthetic pigments, where the major, of course, pigment is chlorophyll, in addition to a group of enzymes and electron carriers that are responsible for achieving the light-dependent reactions and obtaining ATP and reduced NADP. Coming back to the light-dependent reactions, so there are two photosystems. We have photosystem 2 and photosystem 1 in the thylakoid membrane. And along the molecules of photosystem 2 and photosystem 1, there will be a kind of electron flow. And the purpose of that electron flow is to generate ATP and to produce reduced NADP. And we're going to see this in detail over the following slides. So the main purpose of light-dependent reactions is to allow electron flow, and this electron flow occurs along several molecules located on the thylakoid membranes to generate ATP, and ATP will be the source of energy that will be stored in the bonds of glucose, when glucose will be synthesized in the following reactions, which are the light-independent reactions. And meanwhile, these light-dependent reactions do not only generate ATP, but they also generate reduced NADP, which is the reduced form of the hydrogen carrier NADP. Note that in photosynthesis, the hydrogen carrier is NADP and not NAD, and this hydrogen will be utilized for reduction of carbon dioxide into glucose, or in other words, it will be utilized for fixation of carbon dioxide into glucose. We will see this in more detail over the next slides or the following slides. The stages of light-dependent reactions are four essential stages. The first one is the capture of a photon of light, or actually of the energy of the photon of light, by a pigment. And of course, the pigment we are talking about can be of two types, as we said before. It can be chlorophyll, whether chlorophyll A or chlorophyll B, or it can be one of the accessory pigments that we have talked about before. And the function of the accessory pigments is that they widen or broaden the spectrum of wavelengths that can be absorbed by the photosystem to maximize the energy that is absorbed by the photosystem, and finally funnel or converge that energy onto the center of the photosystem, or the main pigment in the photosystem, which is usually chlorophyll. After having captured a photon by the pigment, and the molecule of the pigment will be excited, and this will result in charge separation. Actually, the charge that is separated from the center of the chlorophyll molecule is an electron, and in such a case, the electron will jump to a higher energy level. And when the electron that is revolving inside the chlorophyll molecule, and actually this electron is released from the zinc atom that lies in the center of the chlorophyll molecule, when this electron absorbs a photon and absorbs energy, it will jump to a higher energy level and might leave the chlorophyll molecule altogether. And when it leaves the chlorophyll molecule altogether, this is what we call charge separation. But after the electron has been separated or separates itself from the chlorophyll molecule, we find out that the zinc atom in the center of the chlorophyll molecule has an empty space, which we call, or a vacancy, which we call an electron hole. And this electron hole will be replaced by an electron, as we have said before, from the water molecule. Because as we have said before, the water molecule undergoes photolysis and is split into two hydrogen atoms and one oxygen atom, and the two hydrogen atoms will further be split into two protons and two electrons. So the electron hole in the chlorophyll molecule will be filled in or replaced by the electron that has separated will be replaced by an electron from water. So this is charge separation. Number 3, we have electron transport. This electron that left the center of the chlorophyll molecule will not escape, but it will pass to the electron transport chain, or the series of electron carriers that lie on the inner thylakoid membrane. At the end, we will have a process of chemiosmosis. And in this process of chemiosmosis, we will have transport of hydrogen ions, or diffusion of hydrogen ions down concentration gradient after having been pumped and after having stored a lot of energy. And this diffusion will therefore result in the release of the stored potential energy in the protons, as we will see later on, and the released energy will be useful to generate ATP. Of course, this is a very quick review of the 4 stages of the light-dependent reaction, but every one of these stages will be explained in full detail. And in the light-dependent reactions, there are 2 possible routes for electron flow, because we know that electron flow will result in ATP formation and reduced NADP formation. But in order for this to occur, we should have what we call non-cyclic phosphorylation, which is the usual case in case of light-dependent reaction. So it is the non-cyclic phosphorylation, which is more or less linear, that will result in the formation of both ATPs and reduced NADP. But there is another type of phosphorylation that is cyclic, and we will explain why it is called cyclic. Another type of phosphorylation that will not result in the formation of both ATP and reduced NADP, but will only form one of them, which is the ATP molecule. So every now and then, the non-cyclic phosphorylation stops, and the plant refers to the cyclic phosphorylation that produces only ATP. When does this occur? This occurs when the stores of ATP are depleted and we need to replenish them. So we refer to cyclic phosphorylation that mainly produces ATP. During non-cyclic phosphorylation, which is the usual case, electrons will flow through a series of electron carriers and enzymes that form the electron transport chain. Actually, a series of pigments and then electron carriers and enzymes. First, the electrons will flow through photosystem 2 to produce ATP, and then through photosystem 1 to produce reduced NADP. And as we have said before, the N stands for nicotinamide, which is related to niacin, a vitamin B. Adenine is a nitrogenous base, and dinucleotide phosphate means that this hydrogen carrier, which is nicotinamide adenine, contains, you will never be asked about the structure of reduced NADP, but the structure of NADP is similar to the structure of a nucleotide because it contains adenine, which is a nitrogenous base, and it contains a sugar, and it contains a phosphate, and therefore it is very similar to a nucleotide. We will be seeing and explaining the structure of nicotinamide adenine dinucleotide phosphate later on, but you will never be asked about the details. You just have to know that nicotinamide adenine dinucleotide phosphate is a hydrogen carrier that is produced at the end of photosystem 1, and it is a hydrogen carrier, and it is a product of the light-dependent reactions, and the reduced NADP will be utilized to reduce glucose, to reduce carbon dioxide, to produce glucose. So in non-cyclic photophosphorylation, as we can see on the diagram, the solar rays or sunlight carries photons, as you know, electromagnetic waves, and these photons will excite the electrons in photosystem 2. They will first be absorbed by those molecules that are the molecules of the accessory pigments, and then energy will be transferred from one accessory pigment to another until this energy is funneled onto the chlorophyll molecule or the center of the photosystem. electrons will be, this chlorophyll molecule will be activated, of course, and this results in the escape of electrons from the chlorophyll molecule. As you see, these electrons are immediately replaced by electrons from the water molecule after the water molecule has been split by solar energy. So here we have this famous process of photolysis. When electrons leave the chlorophyll molecule, they are received by a molecule that is called the primary electron acceptor. And then this primary electron acceptor will transfer the electrons to the subsequent electron carriers in the electron transport chain. And we will see that they are not only merely electron transport molecules, but they are also proton pumpers. So these electron acceptors in the electron transport chain, they act as electron carriers. And as usual, they transfer electrons at successively lower energy levels. They are protein in nature, and they are present on the inner thylakoid membrane, but they are also proton pumpers, as we will see later on. At the end of the electron transport chain of photosystem 2 that acts first, it's only called photosystem 2 because it was discovered later than photosystem 1, but in the biochemical pathway, it is photosystem 2 that works first. And then at the end of the chain, of the electron transport chain, the energy that is released from the flow of electrons is used to form ATP. So every time the electrons are transferred from one electron carrier to another, they drop to lower energy levels and release energy, and this energy is used to form ATP. Concerning chemiosmosis, this will be explained later on another diagram. And then these electrons reach photosystem 1, which is at the same time exposed to solar energy and which experienced the same changes that occurred to photosystem 1. So the action center or the center of the photosystem, which is the chlorophyll molecule, lost electrons, and these electrons have been accepted by primary electron acceptors, and electrons are transferred along the chain. And at the end of the day, these electrons, after having reached the lowermost possible energy level, they refind their hydrogen ions or protons and form two hydrogen atoms, and these hydrogen atoms will result in reduction of NADPH. So as we have said on the previous slide, ATP is formed through the electron transport chain of photosystem 2, whereas reduced NADP is formed at the end of the electron transport chain of photosystem 1. One important difference between photosystem 2 and photosystem 1 is the substitution of the lost electrons. Here, when we had an electron hole in the chlorophyll molecule, it has been replaced by electrons from the water molecule. But here, the electrons that have been lost by photosystem 1 upon being excited by the solar energy will be replaced from electrons of the electron transport chain of the photosystem 2 and not from water. So in non-cyclic photophosphorylation, the process begins with photosystem 2. And as you know, photosystem 2 is called photosystem 680 because the center or the chlorophyll molecule that lies in photosystem 2 um, absorbs maximally the wavelength, which is 680. So energy is absorbed by photosystem 680, either directly or funneled by accessory pigments, as you know. And the importance of the accessory pigments, as we have repeated several times, is that they are capable of widening or broadening the spectrum of wavelengths that can be absorbed by the action center. And then electrons from the double bonds in the head of the chlorophyll A molecule. And of course, you know that the head of the chlorophyll A molecule contains double bonds and contains a magnesium atom. These electrons will be energized, meaning that they will be activated by the absorbed light energy. They will jump to higher energy levels. They will leave an electron hole, and these electrons will be captured or received by a primary electron acceptor. Because if they're not captured by the primary electron acceptor, then they will be lost to the environment, or they will return to the original places again, and the reaction would not proceed. And then we have photolysis, and we've mentioned that water gets split by the light energy into 2 electrons, 2 protons, and 1 oxygen atom. The 2 electrons and the 2 H plus or protons are the 2 hydrogens that were present in the water molecule, and the remainder is the oxygen atom. We've mentioned this several times before. And then the electrons that come from water, and here we mean the electrons that come from photolysis of water, will replace the electrons that are lost from chlorophyll A in photosystem 2. So photosystem 2 replenishes its electrons through accepting the electrons that come from photolysis. And then, as we have said at the beginning, that if we have 2 molecules of water, they will split into 2 by 2, 4 hydrogen atoms. But the 4 hydrogen atoms will again split into 4 protons and 4 electrons, plus 2 oxygen atoms, because you have 2 by 1, and the 2 oxygen atoms for O2, and O2 is considered a byproduct, not a waste product of metabolism. We had better call it a byproduct of photosynthesis, and therefore we can say that photosynthesis is the source of all the oxygen that living organisms breathe in on Earth. In the non-cyclic photophosphorylation, and we call it photophosphorylation because, first of all, it is phosphorylation because at the end of the day, you convert ADP into ATP by adding a phosphate. So it is a process of phosphorylation. And at the same time, it is non-cyclic because you do not return to the original site, or you do not return to the original point you have started by. In this electron transport chain of non-cyclic photophosphorylation, which the electrons that left the photosystem 680 will pass along an electron transport chain that will finally form ATP molecules at the end. They will finally form ATP molecules at the end. And this electron transport chain consists of several famous molecules. So we have a plastoquinone molecule. We have a complex of two enzymes called cytochromes, and we have several other proteins. So we're not going to ask you about the details of those proteins. And the electrons that have passed from plastoquinone to the cytochromes to the other proteins, these electrons ultimately end up in the other photosystem 1 to make up for the electrons that were lost by chlorophyll A in the center of photosystem 1. And this is, as I've just said in the previous slide, a major difference between photosystem 1 and photosystem 2. Both have in their centers chlorophyll A molecules, and both lose electrons upon exposure to solar energy and absorbing the energy of photons. But the electrons in photosystem 2 are replaced by electrons from water, whereas those in photosystem 1 are replaced by electrons from the electron transport chain of photosystem 2. The flow of electrons, as we have said, along any of the two transport chains, whether we are talking about the one of photosystem 2 or the one of photosystem 1, but it's obvious in photosystem 2, they are exergonic, as we said before. So with every transfer of electrons from one electron carrier to another, there is energy released. And the cumulative effect of this released energy enables the cell or enables the chloroplast to add ADP, to add phosphate to ADP to form ATP in an endergonic reaction. So we can say that the flow of electrons is exergonic. So every time an electron moves from an electron carrier to another, this electron will release energy, and this energy will be utilized to produce ATP. But the details of how this energy is utilized to form ATP are not that simple. And therefore, we will be studying them on separate slides, or at least on a separate slide called chemiosmosis. But at least at this stage, we can understand why ATP synthesis is called photophosphorylation, because as we said, we are phosphorylating ADP. We are adding phosphate to ADP to form ATP, and all this was made possible because of exposure to solar energy, and therefore it is a process of photophosphorylation that is triggered by sunlight. But how does photophosphorylation occur? Photophosphorylation occurs through a process of chemiosmosis, in which we are going to explain the relationship between the released energy from the electrons and pumping of protons. This picture shows you photosystem 2 on the thylakoid membrane, and you can see the series here of pigments that absorb solar energy. So you have lutein pigment, which is yellow in color and whose maximum absorption occurs at 470. So we call it P470, of course, 470 nanometers. And then we have Z-xanthin, and the wavelength that is absorbed maximally by zeaxanthin. Xanthin is 480 nanometers. In case of the orange, beta carotene, it absorbs maximally at a wavelength of 500 nanometers, whereas the other orange pigment lycopene absorbs maximally at 510 nanometers. And then we have chlorophyll B at 650, and then the center of the reaction, or the reaction center, which is chlorophyll A, and whose maximum absorption occurs at 680. And as we have seen, all these pigments absorb energy, and every pigment transfers energy to the next pigment until all the energy is transferred to chlorophyll A, or actually in other more accurately speaking, every one of these pigments has a different absorption spectrum. So every one of these pigments has a different absorption spectrum, and therefore they can absorb more energy, and all of them funnel or converge their energy onto chlorophyll A, so electrons are lost to join the electron transport chain, and they are replaced by water. In each of these energy transfers, some energy is lost as heat, and this is matching with the second law of thermodynamics. Some energy is dissipated to the environment and increases entropy of the environment. However, and despite these energy transfers that are associated with heat dissipation, yet enough energy will reach the chlorophyll A molecule at the end of the day in order to enable it to lose electrons and to make these electrons reach the electron transport chain. So we have two photosystems, and to the left you have photosystem 2, and to the right you have photosystem 1. If we look at the pigments, lutein, zeaxanthin, beta-carotene, lycopene, and chlorophyll A, we will find that they have, both photosystems have the same reaction center. Photosystem 1 and photosystem 2 have the same reaction center, and chlorophyll A, and have the same types of pigment: lutein, zeaxanthin, beta-carotene, lycopene, and chlorophyll B. But they have a slight difference in the wavelength that is maximally absorbed by the chlorophyll A, and this is not the difference that makes one of the chains form ATP at the end, and the other form NADPH at the end. It's not related to this. Now, what is chemiosmosis? Chemiosmosis in general is the movement of ions across a semi-permeable membrane down an electrochemical gradient. So, for example, when you have sodium ions that diffuse from where they are present in a higher concentration to where they are present in a lower concentration, that is a kind of chemiosmosis. But the chemiosmosis that is worth mentioning here in the case of photosynthesis is the one that helps the formation of adenosine triphosphate through the movement of protons across the thylakoid membrane. Water is present inside the thylakoid, so the protons are generated inside the thylakoids through the process of photolysis. So if you have the grana like this, the piles of thylakoids, and you have the stroma all around, so you first have protons that result from photolysis or splitting of the water that is present inside the thylakoid. But protons are also present inside the thylakoids by being pumped from the stroma into the thylakoid. So we have two main sources for the protons to be present inside the grana or the thylakoids. The first one is the process of photolysis from the water that is already present inside the granum. And the second one is to pump these hydrogen ions by the electron carriers into the thylakoid space using some energy. And we will be talking about the source of that energy later on. We will be talking about the source of that energy later on. But because of the lot of hydrogen ions that are present inside the granum, we can say that the interior of the granum is acidic in relation to the stroma or to the surrounding environment, the external environment of the granum. And there is a famous question that asks about what happens if you prevent pumping of hydrogen ions into the grana. If you prevent pumping of hydrogen ions into the grana, then you are minimizing the concentration of protons inside the grana, and therefore you're making the grana less acidic. In order to be able to the fact of pumping those hydrogen ions into the granum not only causes the inside of the granum to be more acidic, but it also results in the presence of a motive force for the protons, meaning that there will be a higher concentration of protons inside the thylakoid than outside the thylakoids, and this will help diffusion or chemiosmosis of protons from the inside to the outside. So this is what we call the proton motive force. It is a force that helps movement of the protons from the inside of the thylakoid to the stroma, just because protons have been accumulated inside. So pumping of protons by the electron carriers into the thylakoid is what creates that diffusion power or that electron motive force that enables the protons to diffuse from the inside to the outside of the thylakoids. We will stop here and then we will continue next time.